Welcome. The environmental microbiology study the rules of microorganisms in the environment. One of very important concepts, decomposition. It's play a crucial role in the biology of our planet and ourselves. In composition, what we find bacteria break down dead organisms and organic matter, returning nutrients back into the environment. This process is essential for the cycling of nutrients, which is necessary for all living things. The greenhouse effect, this occurs when solar radiation is reflected from the Earth's surface and transformed into heat by greenhouse gas. Greenhouse gas such as carbon dioxide, methane, and nitrous oxide. The change of the climate over the past 150 years, human activities have increased the CO2 concentration in the atmosphere by over 40%. This is changed climate. This has resulted in an increase in mean air temperature, the heat increase, which has broad climate implications. The 2013 Intergovernmental Panel on Climate Change report found that the past century the global average CO2 concentration has increased from 280 to 40 parts per million, and average global air temperature has increased by 0.74. This is a significant number. Moreover, the temperature is likely to increase to 1.1 or 6.4 centigrade in the next 100 years if the tragedy doesn't change, resulting in disasters such as rising sea level, changing ecosystem, drought, flooding, and melting Antarctic sea ice. Research has shown that the combustion of fossil fuels for electricity, transportation, and industry account for over 80% of human produced CO2 emissions. In addition, 60% of the global methane emission comes from human activity, about 70% of which is from industry, agriculture, and landfills. About 40% of the total N2O emission is from human activity, 75% of which comes from agriculture. Soil is the reservoir for chemical elements that form the structure of all organisms on the land. Different organisms utilize and absorb and transform an organic chemical element in different forms to complete the geochemical cycle that support all consumers in the ecosystem, such as animal and human. Most importantly, soil contains chemical elements that are essential to soil organisms, such as C and N. In the most cases, it's believed to be a net sink for atmospheric CO2. However, soil can also be a source of greenhouse gases such as N2O and CH4, especially in an anoxic environment where soil microorganisms undergo anaerobic respiration and fermentation. Soil is a reservoir for chemical elements essential to all organisms and lands. Different organisms use and absorb and transfer inorganic elements to support ecosystem. Soil contains essential elements like C and N, carbon, nitrogen, and is a net sink of CO2. However, it can also emit greenhouse gas like N2O or CH4 in anaerobic environment. Microorganisms in soil oxidize organic carbon using various oxidation. Now we can speak about CO2 cycle or carbon cycle. In aerobic soil, they use oxygen as an electron acceptor, while in anaerobic environment they use alternative electron acceptor. Methanogenesis is a key process where acetate or hydrogen is oxidized to produce methane. Additionally, in aerobic Environment, soil, organic nitrogen can be mineralized with organic carbon decomposition to form ammonium, which is then nitrified into nitrate. In anoxic condition, nitrate can serve as alternative electron acceptor and be denitrified to form N2 and intermediate gases such as NO, N2O. This nitrogen fixation. Certain bacteria can take nitrogen gas from the atmosphere and convert it into a usable form such as ammonia for plant and other organisms. This process is vital for plant growth and food production. Bacteria are used in the production of many fermented foods such as yogurt, cheese, kimchi. They also play a role in the production of bread, beer, and wine. Nitrogen fixing bacteria are organisms that converts atmospheric nitrogen into forms that plants can use. They play a crucial role in the nitrogen cycle, with over 90% of nitrogen fixing being carried out by these bacteria. There are two types of nitrogen fixing bacteria: free-living bacteria, mutualistic bacteria. The mutualistic bacteria form symbiotic relationship with plants as legumes, help them convert nitrogen into ammonia for their growth. Farmers benefit from these bacteria in agriculture by inoculating seeds with commercial culture for appropriate bacteria to ensure optimal growth of legume. Nitrogen fixing bacteria, microorganisms capable of transforming atmospheric nitrogen into fixed nitrogen in organic compounds usable by plant. More than 90% of all nitrogen fixing is affected by those organisms. This play an important role in nitrogen cycle. Two kinds of nitrogen fixing bacteria. The first kind, free-living non-symbiotic bacteria, include cyanobacteria, blue-green algae, anabinia, nostoc, and genera such as Azotobacter, Berkingica, and Clostridium. This second kind comprises the mutualistic symbiotic bacteria. Examples include Rhizobium, associated with leguminous plants, various members of the bee family; Frankia, associated with certain Dictylodinous species; and certain Azosphaerium species associated with cereal grasses. Symbiotic nitrogen fixing bacteria invade the root hairs of host plants, where they multiply and stimulate formation of root nodules, enlargement of the plant cells, and bacteria in intimate association within the nodule. The bacteria convert free nitrogen to ammonia, which the host plant utilizes for its development. To ensure sufficient nodule formation and optimum growth of legumes, alfalfa bean, clover, peas, and soybean seeds are usually inoculated with commercial culture for appropriate Rhizopodium species, especially in soil poor or lacking in the required bacteria. The history of discovering this: Martinus William Perkin, 16 March, who lived from 16 March 1951 to 1 January 1331, was a Dutch microbiologist and botanist who was one of the founders of virology and environmental microbiology. He is credited with the co-discovery of the virus, tobacco mosaic virus, which he called Contagium vivum fluidum. Martinus William was Dutch microbiologist, as we mentioned, and botanist, play biovital role in the field of microbiology. Father of the virology, he was studying disease affecting tobacco plant, tobacco mosaic virus, and found that infectious agent could pass through the filter that blocked the bacteria. This led him to realize there was a new kind of infectious agent, such as a smaller than bacteria, which is termed virus. Enrichment culture also credited with inventing the enrichment culture technique and foundational method in microbiology. This technique allows scientists to isolate and study specific microbes from the environment by providing them with conditions that favor their growth. Environmental microbiology contribute extended beyond virus. He was a pioneer in the field of environmental microbiology, focusing on how microbes interact with their environment. While he is sometimes incorrectly credited with the quote, "everything is everywhere but the environment select," his work on enrichment culture laid the groundwork of this concept in microbial ecology. Nitrogen fixation. This process, diatomic nitrogen gas is converted to ammonium ion and become available to plants, was also investigated by Berkenji. Bacteria perform nitrogen fixation dwelling inside root nodule of certain plants. In addition to having discovered a biochemical reaction vital to soil fertility, agriculture revealed his archetypical example of symbiosis between plant and bacteria. Bejerinic discovered the phenomena of bacteria sulfate reduction, a form of anaerobic respiration. He learned bacteria could use sulfate as a terminal electron acceptor instead of oxygen. This discovery has had important impact on our current understanding of biochemical cycles. Beryllium disulficanes, now known as disulfivibrio disulficanes, the first known sulfate reducing bacteria, was isolated and described by Bejerinic. The carbon cycle. The carbon cycle traces the movement of carbon atom between the atmosphere, living things, biosphere, soil, rocks, and water. When it's living between living things, it's called biosphere, soil, pedosphere, and rocks, geosphere, and water, hydrospheres. Here are the key layers: photosynthesis, cellular respiration, decomposition. Photosynthesis, plant taking carbon dioxide from the atmosphere and use the sunlight to convert it into organic matter. This process releases oxygen back into the atmosphere. Cellular respiration, animal and plant break down organic matter for energy, releasing CO2 back into the atmosphere. Decomposition, dead organisms and organic matter are decomposed by the decomposer, bacteria and fungi, releasing CO2 back into the atmosphere. Fossil fuels, over millions of years, buried dead organisms can become fossil fuels like coal and oil. Burning these fuels release CO2 back into the atmosphere. The oxygen cycle.